Thank you so much for the introduction. I'm really excited to share with you my work on developing safer and more effective cell-based immunotherapies. So we've really entered a new era of medicine that has been completely transformed by cell and gene therapies. We now have the technologies to deliver therapeutic genes, correct mutations, and engineer a patient's own cells to recognize and kill cancer. Around the time I started my PhD, there were emerging news reports about a 5-year-old girl named Emily Whitehead that had undergone an experimental treatment. Emily suffered from a devastating blood cancer called acute lymphoblastic leukemia. Her cancer had relapsed multiple times and was refractory to all the current treatment options. She was essentially destined to die. She enrolled in a clinical trial where her immune cells were engineered in a lab to express a chimeric antigen receptor. These proteins educated her immune cell to seek out and destroy cancer. After the cells were infused, Emily's leukemia almost completely disappeared within just a few days. Doctors said that the therapy wiped out almost 3 pounds of tumor at once. I was really moved by Emily's story and instantly knew that this was the area of research that I wanted to work on. And it wasn't just Emily. Academic clinical trials were seeing unprecedented complete response rates, upwards of 90% in relapsed patient populations. Thousands of people are alive today because of CAR T therapy. So to make CAR T cells, a patient's white blood cells are harvested from blood. They're then genetically engineered using viral vectors to reprogram them with the chimeric antigen receptor protein. They are then expanded to hundreds of millions of cells before being reinfused back into the patient. When I started my PhD, there were no FDA-approved therapies, and by the time I finished, there were 12. This has been a big win for patients, but there are still many challenges and shortcomings. For instance, more than 50% treated with commercial FDA-approved CARs do not actually achieve long-term durable disease control. Moreover, there has been very little success in treating solid tumors, which actually comprise 90% of all cancers. So efficacy and safety barriers are huge obstacles in this setting. So CARs are chimeric proteins that utilize a targeting domain, a transmembrane domain, and intracellular signaling domains. Binding of the CAR to cancer antigens triggers T cell activation, which then leads to tumor killing, proliferation, and cytokine secretion. However, excessive signaling and chronic signaling leads to T cell exhaustion, whereby cells progressively become dysfunctional. Excessive signaling can also induce overactivation of the immune response and lead to toxicity. So for my PhD, I set out to develop a system to regulate CAR signaling with a small molecule to overcome these hurdles. I developed this novel CAR platform turned SNP, whereby I engineered a protease cleavage site between the signaling domains and the transmembrane domain. And I also co-expressed this HCV protease. At baseline, the protease chews up the signaling domains and inactivates the CAR. However, administration of an FDA-approved protease inhibitor inhibits this cleavage event and allows full-length CAR to be produced. Here you can see a Western blot demonstrating high levels of full-length CAR only in the presence of drug, but it is all chewed away in the absence of drug. In this video, you can see SNP CAR T cells in action against green-labeled tumor cells. The SNP CAR T cells are incubated with drug on the left, where they completely annihilate the tumor cells, whereas in the absence of drug, the tumor cells grow out. So these data demonstrate that by regulating CAR signaling, we can control their killing capacity. So next, we wanted to see how SNP CARs measure up to the standard CAR format currently used in the clinic. So we tested them in these really difficult to treat brain tumor models. Here, I injected medulloblastoma cells directly into the cerebellum of mice, then infused CAR T cells by tail vein injection. Strikingly, what we observed is that mice treated with SNP CAR T cells and then dosed with this protease inhibitor, here in blue, had dramatically enhanced potency than the conventional CAR T cells in black. So in this difficult to treat model, the conventional CARs didn't do anything. To further understand the basis for the superior potency of SNP CAR T cells, we pulled T cells out of these mice, stimulated them ex vivo with antigen, and then we observed that SNP T cells retain high functionality and are able to turn on activation markers, whereas conventional CAR T cells simply ran out of steam. Conversely, when we look at exhaustion markers, it is the conventional CAR T cells that display high levels of these inhibitory proteins. So that was the efficacy piece. but we also wanted to test if SNP could be used as a safety switch. So we developed this on-target off-tumor toxicity model, where we built a CAR that targets the ROR1 antigen, which is a tumor-associated antigen, but it also has lower levels of ROR1 expression on healthy tissues. So these CAR T cells will attack healthy tissues and induce toxicity. We treated mice with the SNP ROR1 CAR T cells and gave the drug for 2 days, here shown in blue. We noticed rapid weight loss due to toxicity similar to conventional CARs in black. However, when we pulled drug off, the mice quickly recovered as shown by a rebound in weight, demonstrating that it can be used as a safety switch to reverse toxicity. We initially set out to develop a high-performance, drug-regulated system that we could take to the clinic, because previous systems suffered from leaky activity in the off state, where the T cells are not completely off, and also compromised activity in the on state, where they're not completely on. However, SNP can go from 0 to 100. We compared SNP versus a previous drug-regulated system called DD, or Degrand domains, in the ROR1 toxicity model. The leaky activity of the DD in the off state induced toxicity similar to the conventional CAR. So here there's no drug to turn it on, but it just has this leaky activity in the off state. So this system would not make a very good safety switch. In fact, when I came to check on these mice, I was really struck at how toxic the DD system was in the off state. You can see these poor mice on the left, hunched over and having a difficult time breathing. Those healthy tissues are actually in the lungs that express the ROR1. Whereas SNP, mice looked as healthy as untreated controls. I think these data really highlight that we need clean safety switches, especially when we're testing new CARs or CARs engineered for enhanced potency. And I'm absolutely thrilled to announce that this work served as the foundational preclinical data to motivate a clinical trial of the SNP system in refractory solid tumors. We were actually able to secure funding from ACGT and philanthropic sources, and we hope to open the trial within a year. But to give ourselves the best chance for overcoming the many obstacles of treating solid tumors, next-gen therapies will need to be engineered with multiple enhancements. One just might not be enough. These enhancements could include multi-specificity, resistance to exhaustion and immunosuppression, as well as safety switches. There's much hope that these next-generation platforms that have been developed can be integrated into CAR T cells to overcome these obstacles. However, sophisticated cell therapies incorporating multiple enhancements will need to be engineered with multiple viral vectors because you simply can't fit everything in one vector. And these viral vectors are the only ones that are clinically proven. But engineering cells with multiple viral vectors results in a heterogeneous product that requires purification. But current technologies for doing this at clinical scale would require several sequential purifications for each vector. This is costly, time-consuming, and result in massive cell losses. It's just not practical. So for part of my postdoctoral work, I developed a system called STASH Select. It's a conditionally active selection marker system, which only activates once all the vectors are integrated into the same cell. This system allows for a single-step selection with high purity and high yield. So the core principle of STASH Select is the conditional localization of a selection marker. I fused a protease cleavage site and endoplasmic reticulum retention tag to the C-terminus of a selection marker. This effectively traps the marker inside the cell. However, co-expression of a protease cleaves off the ER tag and allows the marker to travel to the surface. Encoding the stashed marker on one vector, the protease on a second vector, creates an AND gate system where only cells that integrate both vectors express high surface levels of the selection marker, which then can serve as a handle for magnetic-based cell sorting. Here you can see that only cells engineered with both vectors A and vector B have high surface expression for the selection marker. We expanded this approach to a 3-vector system by using a protease that has been split in half. We can then put one half on one vector, the other half of the protease on another vector, and this creates a 3-input AND gate. When expressed within the same cell, the protease subunits reassemble into a functional protease and then send the selection marker to the cell surface. This system would allow for at least 3 different cargos to be delivered to the cells. Here you can see only cells engineered with vector A, B, and C have high surface expression of the selection marker. So as a demonstration of this system, we engineered primary human T cells with 2 vectors, each encoding a different CAR. This dual engineering resulted in a heterogeneous product with only 25% of the cells expressing both constructs, which is the population we want. However, after a single stash magnetic selection, we're able to purify those cells to near uniformity. So we're really hopeful that this technology will help to propel sophisticated next generation cell therapies with multiple upgrades into the clinic. So cell therapies have revolutionized medicine and bring hope to patients suffering from devastating diseases. However, in reality, only a small fraction of patients stand to benefit due to limitations in how these cells are manufactured. Ex vivo manufacturing results in high losses, delayed manufacturing times, and manufacturing failures. Demand has outpaced manufacturing capacities, and there are actually patients dying right now while on wait lists for therapies that could have otherwise saved them. Alternatively, in vivo engineering, whereby a gene delivery vector is directly infused into the patient with the cells engineered in situ, provides an attractive off-the-shelf solution that could broaden access to these revolutionary therapies. However, there are formidable challenges to in vivo engineering, including poor specificity, low efficiency, and the lack of control over the engineered cells. So for my next steps, I'm really excited to develop next generation smart therapeutics that activate within cell types of interest and provide opportunities to control the functions of engineered cells on an unprecedented level. So thank you so much for listening. I'd really like to acknowledge all my advisors, co-authors, mentees, and funding sources and affiliations. Thanks again.